Yeah, thank you so much for the introduction and for inviting me here today. I'm really happy to get the chance to present some of this work on behalf of myself and my colleagues on our validation of mixed strain housing of laboratory mice as a method of reduction and refinement. And so, sec here, there we go. I'd like to start off by just talking about the problem that we're trying to address. And I think Hanno wonderfully introduced this this morning, and I'm going to piggyback a bit on what he said and maybe add a little bit of my own perspective as well. But there's obviously the reproducibility crisis. Everyone knows about that. And in fact, it's been suggested that most published significant, in quotes, findings in biomedical research are likely to be false. And so not only, if this is true, not only does this mean that the literature is filled with misleading information, when this research involves animals, their lives are essentially wasted. And this is a clear violation of the principle of reduction. And regardless of your ethical view on animal research, this should be concerning. And so what are these kind of published significant findings that are false? Well, these are called, perhaps unsurprisingly, false discoveries. And so these happen when a significant finding is not actually real. It shows up as significant in your test, but it doesn't actually reflect anything in reality. And the probability that this happens, that a significant finding is false, is commonly called the false discovery rate. And so this is different than the type 1 error rate of a test. Typically that's, everyone puts that at 5%. That's kind of the commonly accepted. That's what the p-value of 0.05 represents, that cutoff, and the type 1 error rate accepted at 5%. And so what that value is telling you is the probability that any result that you get is a false positive. And that's kind of the accepted threshold through science at large. But the false discovery rate reflects the probability that a positive result is actually a false positive. And so these two values answer different questions. The type 1 error rate answers, what is the probability that the result that I get will be a false positive? But the false discovery rate instead answers the more pressing question: what is the probability that this significant result I obtained is actually a false positive? And perhaps surprisingly to some, the answer to these questions is very rarely the same. And in fact, the false discovery rate is generally much, much higher than the type 1 error rate. And so it's been estimated that when the type 1 error rate is 5%, the false discovery rate can be on average around 30%, which is a bit alarming to me that it could be so high. But so how do false discoveries happen? Hanno touched on a bunch of these. This is related to the reproducibility crisis, but there's a few things that I want to just quickly highlight here. So false discoveries happen because there are institutional incentives for publishing significance rather than truth per se. So when rewards are based on publications of, the number of publications in terms of tenure or grants or things like that, there's a lot of incentives to publish things that are significant and not necessarily true. There are a lot of biases in experimental design, conduct, and analysis, and Hanno went over these very clearly. So this includes things like not randomizing, not being blind, questionable practices like p-hacking and harking, publication bias. Another source of false discoveries has to do with multiple testing. So if you just run a bunch of statistical tests, you're going to find some significant findings just by chance alone. So if you're not correcting for that, that's a contributor to false discoveries. And the one that I want to really focus on today are underpowered experiments. And I think that this is perhaps something that is underappreciated by scientists at large in terms of how they contribute to false discoveries. So, but before I go too far here, I want to take us all back maybe to undergraduate stats class and just give you a refresher on what we're talking about in terms of what statistical power is. And so in the stats jargon, it's the probability that the test correctly rejects the null hypothesis when a specific alternative hypothesis is true. In everyday language that I prefer here, it's basically the probability of detecting an effect that's really there. So if there's an effect there, what's the chance that you're going to actually detect it? And typically, as Hanno mentioned, the kind of gold standard is to get to 80%, although that's very rarely achieved. The power of your statistical test is determined by three main criteria. The statistical significance criterion used in the test, so this is almost always 0.05 or 5%. The effect size, so the difference in the means between the treatment groups, and also is related to the variation within those groups. And the sample size used to detect the effect. This is kind of the common thing that people know about. But I do want to highlight that the configuration, the way that you arrange your sample size also can impact the statistical power of your test, which is really important. And we're going to come back to that a little bit later on in the presentation. And so underpowered experiments, like what's the danger with them? Well, by definition, they have a small chance of detecting an effect. So if your experiment only has 20% power, that means you only have a 20% chance of detecting an effect that's really there. And so this is worrying because you have a very high probability of running a pointless experiment when your power is low. And I think this is kind of commonly appreciated. But something that is more important and much more insidious is that any positive results generated from underpowered experiments are much more likely to be false discoveries. And so how does this happen? I'm going to show you a little bit of a contrived example here just to explain how this can be true. And so this is a kind of a contrived example, but if we say we have our starting population of 200 mice and we're going to apply a treatment, in this case, I've set it up as an enrichment, and we expect our enrichment to be effective in 10% of our sample based on our previous work in our lab. And so in reality, we would see 20 mice improve and 180 mice would not benefit from our enrichment. And when we then translate that over to our statistical test results, if our power is at 80%, we would detect 80% of those 20 mice, which is 16. So those are our true positives, and we would miss 4 mice. So 4 mice would test negative in our statistical test, even though they did improve. Those are the false negatives. On the other hand, we have 180 animals who don't improve, and our significance level of 0.05 means that we have 5% type 1 errors. So we would first see the 171 mice correctly test as not having improved, so that's good. But we have 9 mice that test positive here, even though they did not actually improve as a result of our enrichment. So that's our false positives right there. So when you look at the total number of positives, we have 9 false positives and 16 true positives. So our test is telling us there are 25 positives. But we know that this is not true because we have 9 false positives. And so when you look at the proportion of the false positives to the total number of positives, we have a 36% false discovery rate, not 5% that a lot of people think that we have. And interestingly, as you change the power of your test, so as the power goes down, in this example, if we say the power was actually 50%, the false discovery rate would increase to 47%. So there would be a 50-50 chance that any positive results we get are actually not real. And I've just plotted this out as a graph, as a function, as power increases, the false discovery rate decreases. And so if you have really low power in general, the false discovery rate is very high. And this should be concerning. And I will just note that this exact line is a function of, in this example, the 10% that we expect to be effective. The actual kind of rate of the false discovery is also a function of that value, but the trend is true regardless. And so what can we do about this? There's a few ways that we could increase the power of our experiments. And so, as I mentioned before, there are kind of three key criteria. So we could increase the statistical significance criterion used in our test. 0.05 is, as I mentioned, the universal standard, but it's possible to increase that. Although this is almost never done, as it increases the chance of type 1 errors or false positives. The second thing is, in some cases, it may be possible to increase the effect size within your experiment. So if it's possible, one example is to use a stronger treatment, such as a higher dose. But this is also not always practical given the constraints of the experiment, where usually the treatments of interest are kind of set in stone. And so the third way, increasing the sample size used to detect the effect. This is basically what everyone does. You just add more animals to your study, and you can increase the statistical power that you have. But this has a clear ethical cost, and I think that this is not desirable, just on those grounds. So what if we could use the same number of animals, but in a more efficient way? And this is kind of the question that we ask ourselves here. And so I'd like to take a few minutes here to just explain the theory behind mixed strain housing and how this can help increase statistical power without increasing the number of animals. And so here's a little schematic, an example of what mixed strain housing looks like. So the boxes here represent cages, and the little jelly beans would be mice. Obviously, the different colors are different strains. And then in this example, we also have different colored cages to represent different treatments at the cage level. In our case, this will be standard housing and enriched housing, although it could be any kind of cage level manipulation, such as diet or drug and placebo or anything like that. And so there's the same number of mice in both examples. There's the same number of each strain of mouse. We've just rearranged them so that in the mixed strain groups, there's one mouse of every strain within each cage. And the benefit here, so this is what's known as a split plot design. And the benefit here can be thought of in two ways. The first way is that essentially what we're doing is increasing the number of times that strain is replicated. So on the left, in the single strain groups, every strain is only represented within every third cage, whereas in the mixed strain groups, every strain is represented in every cage. So the number of independent replicates for strain is now 3 times higher. And this kind of benefit does rely on a few underlying assumptions that I want to just quickly mention. And the first is that cage is the correct unit of replication, not mouse. So we're treating that, we're essentially saying that the cage mates are not statistically independent from each other. So cage is a blocking factor when there's more than one mouse per cage in order to avoid pseudo replication. This is really important. And the second assumption is that, as you may know, blocking factors come in 2 types. They're called fixed and random. And in this case, cage needs to be set as a random effect. So that means the analyses will be mixed models. But this is because we're interested in saying something about mouse cages in general, not just about our specific mouse cages. So it makes the conclusions we can draw a bit more broad. And so if anyone is really interested in kind of the math behind this and how this works, I have one more example to show you the benefits here and how split plot designs are effectively more powerful. And so here we have two two graphs. One is for testing treatment. So in our case, this is enrichment. And then on the right, we have the graph showing the way that we would test the strain and the strain by enrichment interaction effect. And what I really want to highlight here is that we'll start on the left in the treatment, but for any given number of cages of animals, in this case, I've highlighted 12 cages, you can see that the critical value of F for the mixed strain design, which is the solid line, is much lower. And so what this means is that the criteria for getting a significant result is lower and therefore easier to achieve. So you're more likely to get a significant result because it's easier to reach this threshold. And these F values come from the ANOVAs and the statistical testing. I don't want to get into the nitty gritty of it, but essentially what we're doing here is lowering the threshold for significance based on lowering the critical F value that you need to achieve a P of less than 0.05. And this is even more stark of a difference when you're looking at the strain and strain by enrichment interaction effects, where the difference is even larger in terms of the threshold being lowered. And also just will point out that this is true for all sample sizes, but it does kind of, the gap between the two designs does narrow as the sample sizes get larger. And in general, you get diminishing returns on the statistical power you gain by adding animals as the sample size gets larger. So that's kind of the theory behind the statistical benefits that you get using a split plot design and kind of how the mechanics of that work. And I wanted to take a quick second to mention some other potential benefits of mixed strain housing. And the first one really is that when your mice look like this, it becomes very easy to tell who is who within a cage. These strains are differentially pigmented. It's very easy to say, you know, this is the black one, the white one, or the brown one. And the benefit really here is that we don't have to do any marking. So this is a report from a PHLASA working group a few years back now, but they looked at kind of the impacts that common marking techniques have on animals, and everything that is in use has at least pain or discomfort attached to it for the animals. It often involves handling and restraint and additional things that we know that are bad for animal welfare. And so by using mixed strain housing, we can just avoid this all completely. The animals just come pre-identified. And the other benefit, and this has been mentioned now a bunch of times this morning, and so I won't hark on this, but increasing external validity. And this isn't exclusive to mixed strain housing, but rather to including multiple strains of mice within your experiment. And this just happens to be kind of the most efficient way that you can do this. And so by including, you know, building this variation into your study population, you can more broadly generalize your results to other situations. And so that is another bonus for using multiple strains. But of course, in theory, everything sounds great, but in practice, we need to make sure that it works. So we tested it, tested this idea to make sure it worked. And so we had a few criteria that we wanted to demonstrate in order to validate mixed strain housing. The first and most important to us was that new welfare issues aren't created, that putting these strains together doesn't make them fight or you know, one strain or individuals kind of monopolizing all the resources or anything like that. So that was really important to us. We also wanted to make sure that the well-characterized phenotypes of these mice are not altered for two reasons. One, we wanted to make sure that mixing the strains together didn't homogenize the mouse phenotypes into kind of a mixed mash of all the different strain phenotypes, so that we could maintain that, you know, heterogeneity that is important. And we also wanted to kind of make sure that as a selling feature, we could ensure researchers that the mice that they're used to working with wouldn't be changed in an unanticipated way as a result of this housing. And finally, we also wanted to double check that the variance in the measured variables is not increased, as this would lead us to losing some statistical power that we kind of theoretically gain from the design. If there's a whole bunch of variance introduced into what we're trying to measure, we don't really realize those benefits. So this is what we were looking for. So we started with a couple of different, so as I mentioned before, we had non-enriched and enriched cages. So on the top here, this was our kind of a standard cage, a shoebox cage with some nesting material and a little house. And on the bottom, this is a picture of one of our enriched cages. So this is actually a typical kind of rat-sized cage that's almost 5 times the volume of the small mouse cages. And it's filled with items that we know that the mice like. So there's a couple of different kinds of running wheels. There's a bunch of different types of nesting materials, tunnels, little paper cups. And their favorite thing, actually, I think, was a little piece of sock there that we cut up and hung suspended from the lid in a hammock. And they really loved to climb up in there. And often they would build their nests up in there. It was very cute. But we set up these trials. So originally we started with 10 cages. We didn't quite end up with 10 cages in every group, unfortunately, but we set them up so that there were some non-enriched and enriched cages for all the 4 groups. So the C57 Black 6 trios, the DBA2 trios, and the BALB-C trios, and then the mixed strain trios. And so we housed them from just after weaning. I think we got them, they were 3 to 5 weeks of age when they arrived. And then we housed them for a total of 20 weeks. And so what did we actually collect here? So we collected data over the period of week 12 to 20. And at the end, we killed the mice by cervical dislocation. And during that time, we collected a lot of behavioral data, a lot of in-cage observations, as well as a few standardized behavioral tests. And these were kind of our bread and butter, looking for welfare issues to see how both enrichment affected the animals, and then whether or not being in a mixed strain trio affected their welfare at all. We also looked at some physiological and morphological characteristics of the animals, and we also took blood samples and sent those for a complete blood count analysis at the end, and looked at all those variables as well. And so in total, there were 26 different dependent variables that we looked at. And we chose these in part because we were interested in some of them, primarily, as I said, the ones related to welfare. But we also wanted to assess a wide range of phenotypic traits in order to look for differences and make the results as applicable as possible across disciplines, so that maybe this would be useful to a broader audience. And so for analysis, we conducted these in JMP10 using this slightly long, complicated ANOVA. But the ones that I've highlighted in red here, these were the terms that we were the most interested in. And so cage type refers to being in a single or mixed strain cage. And then obviously enrichment and strain are self-explanatory. But what these terms represent here and what we're looking for is to ask the questions, does being in a mixed strain housing change how mice respond to enrichments? So do mice in single strain houses respond to enrichments differently than mice in mixed strain cages? And the second term there, we're asking, does mixed strain housing alter strain typical differences? So do we see the same strain phenotypes, whether or not they're in a mixed strain or a single strain group? And these were kind of really important for us, the most important things that we were looking for. So in total, we ran, as I mentioned, an ANOVA for every dependent variable, and this generated 186 different p-values. And so because we were cognizant of multiple testing, we performed a false discovery rate correction for multiple comparisons, which essentially, at the end of the day, lowers the significance p-value from 0.05, in our case, down to 0.0003, so that we can be sure that any results we find, we're pretty confident that they reflect real results. So I'm going to take a minute here just to show you what we found. So first off here, we found we still detected some effects of enrichment, most notably that animals engaged in higher amounts of normal activity and much lower amounts of stereotypic behavior. We also found a whole bunch of different strain differences, as you can see on the bottom square here. And that these were evident, again, regardless of how the animals were housed, because there are no interaction effects. So we still see a lot of main effects, and a lot of these effects are consistent with what we found in the literature. So if you go and look up differences on these variables, for example, in the Jackson Lab mouse phenome database, you'll see a lot of this is kind of in the consistent ranking as would be found elsewhere. And so this was really encouraging that there were no either enrichment interactions and no strain interactions. But of course, how certain are we that mixed strain housing did not affect the data? And it turns out it's really hard to demonstrate a negative because there's always the possibility that we just didn't detect something that was really there. And so the way that we tried to address this concern is by looking at our effect sizes. So we calculated all the effect sizes for all our dependent variables. And here in the table, that's the left column. And these are expressed here as a Cohen's d value, which is just a standardized way of looking at effect sizes across variables. And in this case, these would be considered small effect sizes. So typically, effect sizes on this scale of 0.2 or less would be considered small. They almost all fall into that category. A few of them would be considered medium, and large would be anything higher than 0.8. So we thought we found some small effect sizes, and so we ran some Monte Carlo simulations to ask the question, well, what effect sizes could we have detected given the parameters of our experiment? And those are what I've displayed here on the right. And typically those would be large. So we think that if there were large effects, our experiment could have picked them up. And the fact that we only found very small effect sizes was encouraging. So we're pretty confident that there aren't any major effects of mixed strain housing on any of these variables. Of course, that being said, these classifications of big and small effect sizes are statistical designations and not necessarily biological designations. So some of these effect sizes could be important to certain researchers depending on the nature of their work. So when we look at how variation was affected, here I've plotted the coefficient of variation for every dependent variable for both mixed and single strain. And the coefficient of variation is also just the ratio of the standard deviation to the mean, again, as a standardized way of displaying this information across variables. And so what we actually found across all the variables is that mixing strains actually lowers the variance in the data collected from the animals, which was a surprising result to us. And we don't actually have a good explanation of why this happened. But so we were hoping that there would be no difference, but actually we found an additional benefit here of mixed strain housing in that in this instance, the variation was lowered, which also will help increase the power of this design. And I will note, just as Hanno brought it up a bit earlier, but we also just kind of took a peek at the data and found that in our case, enrichment, the enriched cages also didn't have higher variation than the non-enriched cages, which is also encouraging. And so finally, we wanted to try and quantify the power of the mixed strain design. And we did this in two different ways. And so the first way that I'm going to show you here is that we calculated the power that we had within our experiment in the mixed strain cages and in the single strain cages, and then compared them to each other. And so in this case, I think what we found was that there was no difference in the power to detect effects of enrichment, which may sound, not great, but we compared this using the sample size that we had of 17 mixed strain cages versus 55 single strain cages. So the fact that we're getting approximately equal amounts of power here to detect effects using less than a third the number of cages is actually quite encouraging. The same here is true for the power to detect strain effects. Very similar levels, no difference, even despite the fact that only about a third of the number of cages. And again, also for the power to detect interaction effects. Here it's a bit lower, but not statistically significant difference in terms of the number of cages generating power compared of mixed strain cages compared to single strain cages. And the final way that we wanted to kind of quantify the difference here is looking at the relative efficiency of the design. So we again ran some simulations based on our data to kind of simulate the population that those cages would have been pulled from to estimate how many cages we would need to get 80% power across all of these dependent variables. And so the numbers presented here are medians because there's quite a large range depending on the effect sizes that we found. But what I want to highlight here is that you can see the median number of cages we estimate to get 80% power is drastically lower in the mixed strain models than in the single strain cages. And this is true for enrichments, 252 to 66, for strain effects, 30 to 12, and for the interaction, 282 cages down to 120 cages. So quite the potential for saving animal lives here. So where do we go from here, and what conclusions can we draw? So with this experiment in particular, we think that mixed strain housing represents a refinement to traditional housing practices, at least for female C57 Black 6, DBA2s, and BALB/c mice. We didn't find any negative effects on welfare. There was no increased levels of aggression. Animals never had to be separated. And all of the variables we looked at related to welfare were not increased as a result of mixed strain housing. Importantly, we found that the strain differences were maintained and their typical phenotypes were not affected. So BALB/c mouse still behaved and acted and had the phenotype of a BALB/c mouse, regardless of whether it was in a mixed strain or in a single strain group. As I mentioned, the variance in the data collected from the mixed strain mice was actually lower, although I don't have a good explanation for why that might be. That was a nice bonus for us. And as an aside, this wasn't something that we formally measured, but we talked to our facility technicians afterwards, and they reported that cage side inspections for mixed strain cages were easier, faster, and more reliable, even under red light. It was much easier for them to monitor specific individuals when they can just tell who's who in a cage at a cursory glance. And so overall, thanks to the power gained through mixed strain housing and the split plot design behind it, we estimate that many fewer mice can be used to generate the same or better results. In some cases, less than half the number of animals can be used to generate those results. And so where should we go in the future here? This was kind of a first step, and we don't know, there's a lot of unknowns still. So whether other strains can be mixed, we didn't test males, and this is a big kind of looming question, whether males can be mixed. I know I talked to Brianna Gaskill, and she mentioned she tried males unsuccessfully, and I don't know if that was a result of, the specific strains or what, but it's potentially much harder due to the higher levels of aggression for males to mix them. That's a big unknown. We don't know if this is related to the number of mice in a cage or the age at which they're mixed. So there's some potential to look into that. And I want to stress that we don't want to generalize this to all cases. But what I suggest is that if this is something interesting and possibly useful for you, that pilot studies are very useful for testing this on a lab by lab basis to look more into, your own situation and what can be done. But I do also want to stress, I guess, following Hanno as well, that even if mixed strain housing, isn't for you, I would encourage you to explore more efficient experimental designs like split plot designs or even factorial designs that allow fewer animals to be used while generating better data. And I will stress that you can create split plots without necessarily mixing strains per se. You can mix other genotypes or phenotypes or treatment groups of interest to which a cage level manipulation is applied. And the cage level manipulations don't have to be enrichment. It can be anything like diets or drugs applied. There's like a lot of things that you could do potentially with these improved experimental designs. And so with that, I'll say thank you so much for having me, and I'm happy to try and answer any questions that you might have.